Hello and welcome to BioRules video lessons. Today we are going to be talking about metabolism and metabolic pathways. So, metabolism is the series of chemical reactions that we have in our body that are going to be making we work. We have chemical reactions that build stuff, chemical reactions that break stuff. So all those chemical reactions, building, breaking, whatever you're doing, they all together are going to be called your metabolism. So every single reaction, like cell respiration, photosynthesis, other stuff, they are going to be part of the metabolism. The breakdown of food, all of this, they are all going to be metabolism. Remembering that if a metabolism reaction breaks things, it's going to be called catabolism, from catalysis, But if a chemical reaction builds molecules, builds things, so then it's going to be called anabolism. So metabolism can be catabolism or metabolism. Actually, your catabolic reactions and your anabolic reactions, they're all going to be part of your metabolism. If you want to remember anabolism, just remember the anabolic steroids that athletes use to build muscle. So they are anabolism because you're building. Okay? So for us to talk about chemical reactions, there are many possibilities in our body, and what we're going to be focusing here today goes beyond simple transformation that it was seen in the standard level enzymes reactions. So, but I'll start with the simple transformation first. For example, we have here a substrate like sucrose that we have here in this image in orange, and you have an enzyme in blue. So lock and key model, the substrate sucrose fits perfectly here into the active site of the enzyme, and then when it fits, it puts the water here in the right place to make the break. So then you have products which are going to be here sucrose, fructose and a glucose, and then you have a simple transformation. Like here, as you can see, we have water being put into that molecule, and when we put water, the enzyme is going to be putting the water in the right position. You just break it down. We separate into glucose to one side, fructose to the other side, and we had a hydrolysis. Those are very simple transformation, and they are going to be part of many reactors, especially the ones that are using digestion. For example, breaking down organic molecules that we ate, they are going to use this kind of simple transformation. Sucrose becomes glucose and fructose. Maltose becomes to glucose, and so on. Okay. But however, sometimes some chemical reactions are way too complex to be done by one simple change. For example, changing one amino acid into another amino acid. Amino acids, they can be quite different from each other, and changing from one to another is not so easy as just breaking, separating, adding water, hydrolysis, or something like that. So things are going to be a little bit more complicated, okay? To illustrate this, I'm going to be using an example using words. So not molecules, words. Like if you want to play a game, I want to change the word thread into blink, one letter at a time, rather than one word at a time, okay? If we would change thread to blink, and we would require an enzyme specific to a change, we have 400,000 words in the English language. So we would need 400,000 words to the power of 2 enzymes to make this happen. Because to change thread into blink, we need an enzyme. But to change thread into bread, we need another enzyme. And to change thread into bicycle, another enzyme. So this would be impossible. We would have way more enzymes than we have any kind of reactants to be happening. So our body can't afford having such a complex array of enzymes for all the chemical reactions that can be done. So instead of making a major change, what we can do is to change piece by piece. What if instead of changing thread into blink with one big, huge, gigantic chemical reaction, what if we change it using letter to letter change? So I'll change the thread to another word that is a word, okay, so it's an existing word, but I will change that by only one letter. So thread, oh, I changed to bread. So I changed the T to the B. I need one enzyme, T to B, a TBase. So one enzyme does this trick. Doesn't matter if it's in thread or in tricycle or whatever that you want. If you have a TBase, you change a T to a B. And then I can carry on, for example, changing that A to an E. So I need an AEase. So I change A's to E's, so I change bread to breed. Breed is also a word that exists, and now I have no thread has changed to breed. And then I'll change the R to an L, so I have bleed. And then I change that E, so I can have a second E, NAs, that changes E into N, blend. And then I can change the first E by an I have blind. And finally, I change the G by a K, and I have blink. Instead of making one major gigantic reaction that would require such a complex enzyme and chemical reactions going on to change thread to blink, I change it in several different steps. Thread to bread, then to breed, then to bleed, then to blend, then to blind, then to blink. So I make a series of small chemical reactions. Small chemical reactions are much more likely to be happening for us to have enzymes to do them. It's the way that our body is going to be doing. So instead of changing one to the other, we make this in small steps. We'll call this here, the thread, an initial substrate. It's the first thing that's being reacted. It's a substrate, like we saw in enzymes. Enzymes have substrate, but this is the initial one. And then there in the end, we have the end product. Now, what was produced by the end of this series of reactions is going to be a product, but this is the end product, the final one. All the ones that are in the middle, they are intermediates. Bread, for example, is a product from the reaction from thread, but is a substrate for the reaction to make bread. So the intermediates, they are at the same time substrates for the next reaction, but they are the products of the previous one, so they are intermediates, and usually you don't care much for the intermediates. Okay, a real example on the right, we have the phenylalanine, an enzyme, being changed into fumarate and acetoacetate. One change directly would be impossible, too complex, we can't have that. But we can do that with 6 small chemical reactions. Phenylalanine changed into tyrosine, then change into hydroxyphenylpyruvate, and then change into homogenizate, and then into next one, until you get a fumarate acetoacetate. So we can have several different small reactions. So it's much more likely for that to happen. And that's how our body, and actually anybody, any organism in this planet, are going to be making major transformation in chemicals. Okay, we have what you call a metabolic pathway. Small steps making small changes, one change with one enzyme. So this is what we're going to be having here. So to change phenylalanine to tyrosine, I need an enzyme, which will be a phenylalanine tyrosinase. So it's a molecule that changes phenylalanine into tyrosine. I have enzyme number 1, and then there I have enzyme number 2, enzyme number 3, enzyme number 4, enzyme number 5, and enzyme number 6. So for making this metabolic pathway, I need 6 enzymes. So these 6 enzymes, they are going to be contributing for making this major change from phenylalanine to fumarate acetoacetate. 6 of them. So it's not one reaction, it's a cascade of reactions. Okay, and they're going to be calling this a metabolic pathway, because it's like a path, like a trail in the jungle. So you have a pathway that you have making metabolic reaction. In this case here, I'm making small changes to make phenylalanine to become fumarate and acetoacetate. Okay, it requires many enzymes, that's a fact. Okay, you need one enzyme for each single step. So every single step here, you need to have one different enzyme. But you have an initial substrate, and you're going to have an end product in this reaction. So that's what's going to be matter, because your body usually wants the initial substrate, and usually it wants the end product. They don't care much for the intermediates. Intermediates are part of the way to reach a goal. So your body wants fumarate acetoacetate, breaks phenylalanine. Don't want fumarate acetoacetate, keeps phenylalanine. Usually you go, you start, and then you have your production out there. Okay, Metabolic pathways, they may be chains, like what you have here, one after the other, making a little chain, or they may be cycles. So let me give you an example here of a cycle. This here is part of a very important cycle that we are also going to study called Krebs cycle or citric acid cycle, where we transform an acetyl 2-carbon molecule and we break it down, releasing energy. We put carbon dioxide out, it's part of the respiration, and in that process we make little packages of energy that are going to be these guys over here. We are going to be making this little piece of energy. So it's a cycle. And then goes back to what it was over here. And this here receives this acetyl again and goes on. So we have a metabolic pathway. That's not a pathway, it's a metabolic cycle. This here is a chart illustrating metabolic pathways in an organism. So this here is a very, 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 very complex one. Look at this. Many chemical reactions. And here, if you see, we are going to have in the middle many cycles here. Look at that. We have pathways, we have cycles. Krebs cycle actually is part of this. But you don't need to bother yourself much with this in here. Okay, having said that, I hope you understood. If you didn't, go back into the video, now take a look again. The idea of metabolic pathway, many enzymes, one after the other, all that stuff. Controlling chemical reactions is also going to be part of our concerns when you talk about metabolic pathways or even simple reactions. Our reactions should be controlled. If they are not controlled, we would explode in a big ball of fire of chemical reactions. So what we have here is a way of making your reaction to start or to stop when you want. So basically, chemical reactions in your body, they should start to stop under command. So for that, there is a process called inhibition. Quite simple, quite straightforward sometimes. And let me show you here how to control the chemical reactions in your body, okay? Basically, how do you turn on and off a chemical reaction? And if you have enzymes that are making a chemical reaction to happen, one way that you probably would think is that we could break the enzyme and destroy it. If you destroy an enzyme, the chemical reactions are going to be very unlikely to happen. So if we break this here, oh look, there's no blue enzyme here anymore, no sucrase, because this is a sucrose being broken, we don't have chemical reaction anymore. Yes, and that's true. No enzyme, no chemical reaction, that's one way of controlling things. But building enzymes and breaking enzymes is a very expensive business. Your body cannot afford breaking and building enzymes every time it wants to regulate a series of chemical reactions in your body. So what are going to be happening? So let's see this chemical reaction that was just broken over there. So the sucrase broke the sucrose, separates into fructose and glucose. No, previous classes that we had, those molecules that I told you that you should start getting used to it. And now we have there, it was broken and the substrate went away. And now we have more substrate going on, making product going away, substrate going in, product going away, substrate going in, product going away. So basically, if you have substrates, the enzymes are going to make them react. So if you have a substrate, the enzymes are going to be making the reaction happen, building more and more product. But here, there's a catch. If you start having so much glucose and fructose around, and they are not being consumed, so basically you start having way too much, your body should say, okay, maybe I don't need so much glucose and fructose, maybe I should stay with the sucrose, the initial stuff. So, because if you have too much of something, I don't need that, so let's make it stop. But instead of destroying this enzyme, there's another clever way, simple way, that this chemical reaction gets stopped. Basically, by chance, molecules bounce into the active site. If this is the substrate bouncing the active site, you have a reaction and you produce the product, the glucose and the fructose. But what if a glucose or a fructose, the products themselves, hit the active site? If they hit the active site, then you have a blocking, because they fit there. They just got out from there. And if they are in there, no other substrate can go inside. Okay? If one of the products are there, just by chance bouncing, they occupy the active site, so no sucrose will be able to enter and to hit over there and to be acted upon by the active site to be hydrolyzed. So in this case, the chemical reaction is going to be stopped for a while that glucose is over there, by competitive inhibition. The inhibitor, in this case the products it's themselves, glucose or fructose, they are going to be occupying the active site. So they compete for the active site with the substrate. With the inhibitor, glucose or fructose in this case, the substrate, sucrose, cannot get in. But after a while, this glucose starts being used, the fructose starts being used, and they bounce out as well, because they bounce in, they bounce out. And if you have more substrate than product, then you have a bigger chance of this enzyme to go inside, bounce in there, and then bounce outside again. Okay? So this here is inhibition, competitive inhibition, and it's usually very useful when you have a simple reaction, like sucrose becoming glucose and fructose. That's it, one reaction, one enzyme, things works, they work wonderful like this. Okay? So, competitive inhibition, it states that the more inhibitor you have, so in that case, the more product you have, higher the chance that they will hit and occupy the active site. So the higher the inhibitor, more likely that they compete, they don't allow the substrate to go in, so it's off. So it's not only a matter of on and off, it has to do with how much you have. Too much substrate, a little bit of the product, the inhibitor in this case, chemical reactions go on, more likely for the substrate to hit the active site. Too much of the product, in this case the inhibitor, and not so much of substrate, much more likely for the inhibitor to hit the active site and for the reaction not to happen. So slow down the reaction. So we are going to have this kind of control. No, when the inhibitor lows down, then you have more substrate and then the reaction is on. Too much product, too much inhibitor, off. Too much substrate, so not much inhibitor, the reaction is on. This here is the simple competitive inhibition, very common in simple reactions. But what if we want to inhibit a metabolic pathway? Inhibiting a metabolic pathway is tricky because, if we're going to be using competitive inhibition, is it possible? Yes, it's possible. If you have too much fumarate or acetoacetate, what is going to be happening? They will competitively block enzyme number 6, which will end up accumulating for fumaryl acetoacetate. They accumulate intermediate. And then too much fumarate acetoacetate starts to build up because this reaction stopped. And then they start blocking this chemical reaction here that produces too much methyl acetate. And then too much of this would start blocking this enzyme and accumulating this. And then too much of this blocks there, and then these ones accumulate that one. And then too much of this blocks that one, and then accumulates this one. And then finally, too much of this blocks this chemical reaction, and you have too much phenylalanine. The problem with this is that takes a while. You need to have too much of the last one to then start building the other one, and then you build a lot of intermediates in the middle of the way. Man, this is a mess. This is not supposed to happen. So no, shouldn't be like that. Okay, what's going to be happening is that there is a clever way, a smaller way, because usually our body wants the initial substrate or the end product. We don't want the intermediates. Initial products, okay, we keep them there. End products, okay, if you need them, we know you want them there, but not everything in the middle, especially if you have a very long cascade of chemical reactions. So in that case, there's a different way of blocking a chemical reaction. Okay? No good in accumulate intermediates. Beautiful. So how? Let me show you. If it's not going to be like this, fumarate acetoacetate, which fits in enzyme number 6 because it just got out from there, is not going to be the solution. What would be the solution? If you fought on this, that's the solution. The end product accumulation should inhibit the first enzyme. If you stop the first enzyme, you stop everything. So now, if fumarate or acetoacetate stops the enzyme number 1 to break phenylalanine into tyrosine, now we have the entire chemical process stopped. However, no, final product blocking over there. Okay, sorry, not however. No, it's blocking the first enzyme and make things to be stopping. But the problem is, however, but the problem is this cannot be competitive because fumarate acetoacetate now is too different from phenylalanine or tyrosine, the two things that fit on enzyme number 1. Phenylalanine is the substrate, tyrosine is the product of enzyme number 1. The lock and key model, the induced fit model, all of them say that the enzymes are specific. So fumarate acetoacetate do not fit into enzyme number 1. So no, not competitive. Okay? End product is so different now, has been changed so much by so many other enzymes in the middle with many chemical reactions that they don't fit the active site anymore, so they cannot compete with phenylalanine, so they cannot hold down this chemical reaction. So what are we going to be doing then? So how? You're still right, this is the way. We should use the last one to stop the first enzyme. So for this case here, there's one kind of enzyme that have evolved that makes it's an allosteric enzyme that makes an allosteric inhibition. Allo means away, so allosteric is when an enzyme has an allosteric site, so it's another site rather than the active sites. Here we have active site, it fits the substrate, it could fit as well the products, very good, but this enzyme also has another site here in this case here on the bottom of this enzyme called allosteric site, and this place actually fits a chemical, fits an inhibitor. And if this inhibitor fits in there, we have here, now I'm painting it in black, here you have an inhibitor fitting in there. This changes the entire protein and changes the shape of the active site. You see that the active site now is twisted, it's locked, and now the substrate cannot go in. The enzyme is blocked. So it is an inhibition. The inhibitor here ends up blocking the chemical reaction, but not by competition, but by changing the enzyme itself to make it not to fit the substrate anymore. This reaction is blocked. Okay, so there's the allosteric inhibitor. Usually it's going to be the end product. So in that case, what we had before, fumarate or acetoacetate. Okay, we have a change in the enzyme three-dimensional shape because they have it over there, and now the active site doesn't fit the substrate anymore. And if they don't fit the substrate anymore, the chemical reaction is stopped. This is allosteric inhibition. Just to show you an example, here on the top we have a chemical reaction happening just by fitting the active site over there. They react, release the product, beautiful. When you have an allosteric inhibition, this inhibitor here, when they fit on the allosteric site, the non-competitive site, they fit in here, they change the shape. And once they change shape, the substrate cannot go there anymore, and the chemical reaction is stopped. Okay? Another example here now with a metabolic pathway. This enzyme number 1 breaks triangles into rectangles. The enzyme number 2 breaks rectangles into this weird wave-like rectangle. And the enzyme number 3 changes this wave-like rectangle into a square. And the square, by time, comes here and connects to the allosteric site of enzyme number 1. And once it's connected there, it changes the active site. And now with the active site being changed, now the substrate cannot fit anymore, and then you stop the reaction. So if this here stops, this here stops, this here stops, and then you blocked the entire metabolic pathway. This is allosteric inhibition. Okay, so the allosteric inhibition in a metabolic pathway usually, okay, the end product inhibits usually the initial enzyme. They fit in the allosteric site, they change the active site, and then the reaction stops. Okay, very good. So this here is a graph illustrating how the concentration of substrate influences enzyme activity. You might have seen, if you didn't, then maybe this part here, okay, this part might be a little bit more confusing, but if you didn't see, you should have seen that when we increase substrate concentration, we increase enzyme activity. So if you have no substrate, we have no activity. There's nothing to be broken. The more substrate we have, more activity we have, the more. However, this is not linear going up all this way forever, because there's a moment that you will start having so much substrate that you might not have enough enzymes to be breaking them down. Basically, all the active sites might be occupied. There will be a moment that you have so much substrate that now you reach a limit, and you reach actually a maximum. You are breaking things as fast as the number of enzymes you have to break goes. We saw this here, standard level, the effect of substrates into the enzyme activity. Now, what happens if you have different kind of inhibitions? So if you have different inhibitions, for example, competitive inhibitor, the more substrate you have, more substrate in relation to the inhibitor you end up having. If you have, for example, 1,000 enzymes, and you have there 500 inhibitors, and you have 0 substrate, no reaction happens. I put one substrate, I have one substrate to 500 inhibitors, almost nothing is going to happen. But if I start to increase this up, when I get to 500 substrates and 500 inhibitors, now I'm increasing the substrate concentration here, I already have half and half. So the chance that an enzyme will work is going to be basically half and half, 50%. I got to 50% of the chemical reaction. So I'm already getting here. And then if I start having more, if I have 1,000 substrates to 500, I have 2 times the chance of the enzyme to work to 1 time of it not working. If I have 1 million substrate to 500, then basically I have so much substrate that the inhibitor don't count anymore. So ultimately, competitive inhibition, they are going to be reaching the point that basically they will get practically to the same level of the reaction without inhibition in very high substrate concentration, because at a certain point the inhibitor doesn't count anymore if you have way too much substrate. On the other hand, non-competitive inhibition also blocks the chemical reaction. If you have the inhibitor, you're going to be having in the beginning basically the same stuff as you had with the competitive inhibitor. Now you have the inhibitor over there, they're blocking enzymes. It's as if you had less enzymes. So you have less reaction. You start having more substrate, you have more reaction, but not so much as if you had no inhibition. But there's a catch here. If you have competitive inhibitor, they are going to be blocking enzymes. So the relationship is not with the number of substrate. I can increase substrate, increase, but if I have non-competitive inhibitor, enzymes will still be blocked. So basically the non-competitive inhibitor, they levels down at a lower level of reaction. Basically because one non-competitive inhibitor, allosteric inhibitor, they take out an enzyme from the equation rather than fighting for an enzyme with a substrate. So more and more substrates make no change because you have basically less enzymes available for the chemical reaction to go. This is a classic example of how the competitive and non-competitive inhibition work in relation to how much you have of them around. So this will be quite important. We might have another discussion, a deeper discussion on this later. Okay, so the maximum velocity with very high substrate concentration for the competitive inhibition is practically the same as without inhibition, because in the end, too much substrate means that the competitive inhibition makes no difference. But the non-competitive inhibition will end up having, no matter how high the substrate concentration is, a lower maximum velocity, because you end up reaching a certain number depending on the amount of enzymes that are available at that point, and with the inhibitor you have less of them available. Okay? Just to take advantage here, negative feedback is a way for us to control this. What we are talking actually for the moment here is negative feedback. This will be very, 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 very important for many things in biology. Actually, this is important for many things in biology, in physics, and chemistry. But it's very clear a negative feedback here when we have a metabolic pathway, when you have a chemical reaction, and we have an inhibitor. Basically, if you have phenylalanine becoming fumarate acetoacetate, you end up accumulating fumarate acetoacetate. So if the pathway produces much end product, you end up having way too much end product, way too much. What is the reaction that has to be done? Hey, slow this down. So this enzyme, the enzyme number 1, then is blocked by fumarate. Okay, so the first enzyme is blocked, the pathway shuts down. If the pathway shuts down, now you reduce the amount of product there in the end. So too much product make it react to lower it down. That's just what happened over here. But then, if you blocked over there, you start having too little product. So too little inhibitor. So if the end product reduces, we are going to have less end product inhibition. So we are going to stop inhibiting over there, and then the cascade starts, and then you start making more. So too little, make more reaction. Too much, make less. Too much, make little. So basically, negative feedback is called negative because it goes in the other way around. If you have too much, make it go down. Too much of this, make it go down. Stop here and make it go down. Too little, let it go, and they'll produce more of the product. So basically, it's a reaction of too much, reduce, too little, increase, and that's it. Okay? If you have more product, you have less reaction. If you have less reaction, you have less product. But if you have less product, then you're going to have more reaction, then you have more product. So the less, the more. You react in the opposite direction, negative feedback. We are going to have a very good discussion on this because it's a lot of information. I hope you have got the basic idea and you get prepared for the class, because this class is going to be of very good discussions. Make your notes, get anything that you didn't understand and bring to the table, so you can make sure that you are understanding this very high level concept of metabolic pathways, its inhibition, negative feedback, and everything surrounding it. Okay. Now you have a questionnaire. Go there into the website or use this QR code to find it out. Do the questionnaire to see how well you have understood this, collect your doubts, and bring to class. I see you in another time.